Welcome to Module 4 of our GMP series. Today, we are diving into Subpart E, Control of Components and Drug Product Containers, and closures of 21 CFR Part 211. This section is critical because the quality of a finished pharmaceutical depends on the integrity of the materials and the vessels that protect them. We begin with the high-level life cycle of a material, which moves through 7 distinct stages, from receipt to final issue. Every step must be governed by written procedures. The Quality Control Unit acts as our gatekeeper, ensuring nothing moves to the next stage without formal verification. Upon arrival, the first line of defense is a thorough visual examination for labeling, damage, and contamination. We establish traceability by assigning a unique internal lot number. The material must be immediately identified as quarantined. Proper storage is a regulatory requirement. Components must be stored off the floor and spaced to allow for cleaning and inspection. We must also maintain environmental controls. A simple rule is, if you can't clean around it or inspect it, it is stored incorrectly. Quarantine is our yellow light. Materials must stay in this status until formally released by the Quality Control Unit. This can be achieved through physical means, like a locked room, or through a validated electronic inventory system. Once in quarantine, we move to sampling. We must collect representative samples using aseptic techniques to prevent contamination. If testing for stratification, do not mix samples. Every sample container must be meticulously labeled to maintain the chain of custody. Testing is where we verify claims. Identity testing is non-negotiable. At least one specific test must be performed. We can rely on a supplier's report of analysis for purity and strength only if we perform the identity test and validate the supplier's reliability. Beyond chemical identity, we look for physical and biological adulterants. Microscopic examinations detect filth or infestation in susceptible lots. If a material has the potential for objectionable microbiological contamination, it must undergo rigorous testing. Testing culminates in a binary decision, approval or rejection. Released material meets all specifications, and its status changes to released. Any failed material must be quarantined to prevent accidental use, eventually leading to its destruction or return. Once material is approved, we follow the first in, first out FIFO principle. This rotation ensures the oldest approved stock is used first to prevent degradation and expiration. Temporary deviations are rare and permitted only when necessary, such as for validation studies. Quality isn't static. Materials must be retested if stored for long periods or exposed to adverse conditions like heat or air. If degradation occurs, the Quality Control Unit must nullify the previous approval and reject the lot based on the new results. Containers and closures must be chemically neutral, meaning non-reactive, non-additive, and non-absorptive. The system must also protect the drug from light, moisture, and temperature fluctuations throughout its shelf life. The cleanliness of containers is paramount. All must be sanitized. For injectables, sterilization and validated depyrogenation are required. These cleaning methods must be written, standardized, and strictly followed. To summarize, the control of components and closures rests on 5 pillars: written procedures, clear chain of custody, verified identity every time, inventory control, and the final authority of the Quality Control Unit. Thank you for subscribing to my GMP training on YouTube. If you want customized training for your company, please reach out via email.